timeline, as defined by the Merriam-Webster Dictionary, is the list of important events for successive years within a particular historical period. Now, as we integrate the sciences of biology and chemistry and technology, we come up with making a well-curated timeline dedicated for our degree program, Biology, presenting our timeline of genetic engineering. To start off, genetic engineering is a process by which genetic material of chosen species is changed in such a way in order to come up with possible production of new substances and functions as well. In other words, it is called genetic modification as it is stated on the slide. Paul Berg is considered to be the father of genetic engineering. Now, let's traverse our way to the timeline of genetic engineering. First, I think even non-biology students know this. In 1953, James Watson and Francis Crick discovered the structure of the deoxyribonucleic acid, or what we call DNA. We take credit where credit is due, so it is right and just to mention that this groundbreaking discovery of Watson and Crick was possible because of the decades of study pioneered by Friedrich Mischer that started in the 1860s, and the contributions as well of Phoebus Levine and Erwin Chargaff. In this discovery headed by Watson and Crick, they identified the DNA as a three-dimensional double helix structure that stored genetic information. Next up, in the year 1973, Herbert Boyer and Stanley Cohen developed genetic engineering by inserting DNA of one bacterium to another one. This contribution led to the very first GMO, or what we call the genetically modified organism. With this breakthrough, it opened up the doors of bacteria development in order to produce human insulin for diabetic patients, which in 1982 became the first consumer GMO product approved by the U.S. Food and Drug Administration. Speaking of genetic modifications, in 1974, Beatrice Mintz and Rudolf Jaenisch created the first genetically modified animal. This was a collaboration of knowledge between the virologist Jaenisch and the mouse embryologist Mintz. As they utilized their knowledge with their specializations, they demonstrated the possibility of modifying mice genes by injecting a DNA virus, so-called the SB40 virus, into the early stage mouse embryos. With this work, it led to the creation of genetically modified transgenic mice.
So, in year 1977, Frederick Sanger and his colleagues developed groundbreaking DNA sequencing method, and is also known as the Sanger sequencing or the deoxysequencing method. So this method revolutionized the field of genetics by allowing for the determination of the precise order of nucleotide bases in DNA molecule. The Sanger sequencing method allowed the scientists to read DNA sequences more accurately and efficiently than previous methods, making a significant advancement in genetics and molecular biology. This method plays a crucial role in sequencing the human genome and continues to be used in various applications and also in sequencing projects. So next is the development of the first transgenic tobacco plant in 1983, and it was a landmark achievement in the field of biotechnology and genetic engineering. This breakthrough marked one of the earliest successful attempts to introduce foreign genes into plants' genome, opening the door to the creation of the genetically modified organisms for various purposes, including agriculture and scientific research. So this achievement was a key step in the broader trajectory of genetic engineering, contributing to the development of the genetic modified crops and the ability to manipulate the plant genetics for specific traits like pest resistance, improve yield, and enhance nutritional content. Next is the discovery of the polymerase chain reaction or PCR in 1985 by Gary Mullis. It was a transformative breakthrough in molecular biology and genetics. PCR is a widely used technique that allows scientists to amplify and replicate specific segments of DNA, making it an essential tool in various fields including genetics, forensics, and biotechnology. And the significance of discovery of the PCR and revolutionized molecular biology by allowing scientists to quickly and precisely replicate DNA in the laboratory. PCR has numerous applications, including DNA sequencing, genetic testing, forensic, disease diagnosis, and the study of ancient DNA.
CRISPR, short for clustered regularly interspaced short palindromic repeats, is a revolutionary genetic tool. It simplifies gene editing by enabling precise modifications to DNA. This breakthrough technology allows scientists to make subtle alterations in DNA strands, opening up new possibilities in fields like medicine and agriculture with potential implications for treating genetic diseases, creating genetically modified organisms, and advancing our understanding of genetics. In 1990, French Anderson and his team at the NIH achieved a milestone by conducting the inaugural authorized gene therapy procedure. They administered this groundbreaking treatment to a 4-year-old girl afflicted with severe combined immunodeficiency, or SCID. The Human Genome Project accomplished a remarkable feat by sequencing more than 90% of the human genome. This monumental endeavor provided a comprehensive understanding of the genetic blueprint that defines our species. It paved the way for numerous breakthroughs in genetics, medicine, and biotechnology, offering insights into the intricacies of human biology and the potential for advancements in personalized medicine and disease research.
And George Church are the scientists who created multiplex of the genome engineering or MAGE, and it was discovered or developed in the 2000s. MAGE is a groundbreaking genome editing technique that allows scientists to make multiple changes to an organism's DNA simultaneously, and it greatly speeds up this process of genetic modification. MAGE significance lies in its ability to accelerate genome editing, making it faster, more cost-effective, and precise. This technology has opened doors to various applications such as developing medicines, producing biofuels, and studying genes related to disease, and it has the potential to revolutionize healthcare, agriculture, and environmental science, offering new ways to improve human health in the world we live in. The development and research surrounding artificial chromosomes involve contribution from various scientists and researchers over the time, and it is not attributed to a single proponent. Artificial chromosomes has been a subject of study and development over several decades with significant progress made since the early 1990s. Artificial chromosomes synthetic DNA structure created in the laboratory to mimic natural chromosomes. They can carry and replicate genetic information like real chromosomes and are often used as a tool in genetic research and biotechnology. Artificial chromosomes have a profound impact on genetics and biotechnology, so their significance lies in their ability to carry and manipulate large segments of genetic information. Research uses artificial chromosomes to study gene function, development, genetically modifies organisms, and potentially that genetic disease have the potential to revolutionize gene therapy and contribute to their understanding of genetics and human health. Tal effector nucleases or TALEN is a genome editing technology that has been developed by various researchers in the field of molecular biology and genetics. It's not attributed to a single proponent, and TALEN effector nucleases or TALEN technology emerged in the early 2010s, and TALEN is a genome editing technique that uses custom designs proteins called TAL effectors to target specific DNA sequences. These proteins are engineered to bind to a particular DNA sequence, allowing to precise gene editing. Talents are significant because they offer a highly specific and versatile method in DNA, and they have used in various research and medication, including the development of potential treatments for genetic disease. And also, talents have the potential to advance gene therapy and genetic research, bringing us closer finders, cures for inherited condition, and improving our understanding of genetics.
the discovery of the CRISPR-Cas9 in 2012. In 2012, Jennifer Doudna and Emmanuel Charpentier made key discoveries in the field of DNA manipulation with the CRISPR-Cas9 system, the so-called genetic scissors, which led the women a Nobel Prize Award in Chemistry in 2020. Clustered regularly interspaced short palindromic repeats with associated protein 9, or the CRISPR-Cas9, is a unique technology that enables geneticists and medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequences. Scientists today use it because of its inexpensiveness, versatility, accuracy, and it is customizable. The CRISPR-Cas9 has since been a tool for solving issues such as eradication of malaria and influenza pandemics that helped save many lives. First edited human embryo using CRISPR-Cas9. In 2015, due to the CRISPR-Cas9 being cheap and easy to apply, a group of Chinese researchers were the first to use the CRISPR to edit the genomes of non-viable human embryo. Due to this, it was theorized that it was possible to create a genetically modified baby through in vitro fertilization. This brings us to the last event in our timeline: the attempt to use gene editing on human fetus in 2018. Following the 2015 event of researchers using CRISPR-Cas9 on human embryo, another Chinese scientist, biophysicist He Jiankui, along with a team from Southern University of Science and Technology in Shenzhen, China, recruited couples in an effort to create the first gene-edited babies. The CRISPR babies would not have the gene CCR5 in hopes to create an offspring that is resistant to HIV, smallpox, and cholera. It was reported that 8 twins were born from one of the couples and are doing well right now. But the event still remains under questions and is referred to as the gene editing scandal. In relation, the Third International Summit on Human Genome Editing in March 2023 talked about the regulations in China following the 2018 misuse of heritable human genome editing, because they said that the practice was unethical. A timeline helps us to trace back to a certain point in history to see how the world today is affected by the happenings in the past. It is important for us to have knowledge of the past in order to move forward, using the mistakes and achievements of past discoveries and experiments as our guide to continue to provide for a better world. Let us all be hungry for knowledge and know more about the science of life.